Hi, I'm Dan Crane. I'm a professor in the biology department here at Wright State, and in this video I'm going to talk about how it is that statistical weights get attached to single source DNA profile samples. All the slides that I'll be using today will be available at www.bioforensics.com, as well as a number of additional materials that you might find helpful if you want to wrap your head around these issues even more than I get into them here in this video. But with that said, let's get right to work and start talking about statistics as they apply to DNA profiling. All right, so let's get this out of the way right out of the gate here. A 13 locus DNA profile match is exceedingly rare. All right? The chance of a coincidental match between two randomly chosen unrelated individuals is vanishingly small. But nonetheless, in the United States as well as abroad, there's an expectation that for any DNA profile match that a statistic actually be attached. It's not enough to simply say that the profiles are rare. We need to actually generate a specific weight to that DNA profile match. And there are things that can complicate attaching a statistical weight to DNA profiles, things such as the fact that the sample is a mixture, it includes relatives, or the information in the sample is incomplete. But those are complications that we're not going to get into in the course of this discussion. That's for a later video. Here we want to talk just about, in the best case scenario for a single source sample where all the information is present, what's the chance that a randomly chosen person would have the same DNA profile we're seeing in an evidence sample. So, what does a DNA profile look like? Well, that's something that's been discussed at some length in another video as part of this series, but the long and the short of it is this: DNA profiles are essentially a set of peaks and valleys that you see associated with graphs. This particular slide is showing you a reference profile, the DNA profile that's been generated from an individual's reference sample. And to a large extent, a DNA profile can be reduced down to a series of numbers. These are the names of the alleles that are seen at each of the loci that have been tested. And you can see different test kits can look at a variety of different loci. Here we're looking at a total of 15 different polymorphic loci, and for each of those loci, we get some information about which alleles are present. Again, taken together, that constitutes a DNA profile. But where we want to go with this particular discussion is not so much about what it is a DNA profile looks like, but we want to start talking about how it is those DNA profiles get interpreted. And so this next slide gets us set up to do just that. What I have for you here is an evidence samples electropherogram. And the question at hand for any DNA profiling test is who might be included and who might be excluded as a possible contributor to that evidence sample. So again, we have an evidence sample here, but at the heart of it all, we're going to need to do a comparison. And what should we compare to? Well, the DNA profile that comes from a suspect's reference sample. And to the right now, you can see appearing there such a DNA profile. This is a suspect's DNA that's been tested with the very same kit that the evidence sample has been tested with. And the question before us right now is, could this suspect be the source of the DNA that we see on this particular evidence sample? And I tell you what, you don't need any special degree. There's no special class you take in graduate school when you're attending a university that tells you how to do this. It's pretty straightforward stuff. What I'd encourage you to do is just look carefully at the evidence sample's DNA profile and look to see what you find in the suspect's profile at the same corresponding positions. So if we look in the upper left here, the very first locus in blue is one that has the name D8. And what do we see in the evidence sample but a 12 and a 15 allele? That's the DNA profile we found for that locus in the evidence sample. And it's not hard to tell, just at a glance you can see that at the very same locus, D8, the suspect that we're considering here has a 14, right? In some sense, we could call that a 14-14. The individual is a homozygote. He has two copies of the 14 allele. But I think more to the point, he doesn't have a 12 or a 15. So right out of the gate here, we can say with some confidence that there's a problem if this individual is the source of this DNA. The problem is where's this 12 and 15 coming from? And further, where's the 14? So at the very first locus, we can start to get the impression that suspect 1 is not the source of the DNA on this evidence sample. And at each subsequent locus that you look at, and again, there are many that you could choose to look at here, we find time and again that the suspect can't be the source of that evidence sample. So what do we say? Well, in one word, what we say is suspect 1 here is excluded as a possible source of that evidence sample. And there's really not much more that needs to be said. Quite simply, he's excluded. And in fact, there is nothing more that needs to be said. We don't need to attach a statistical weight to an exclusion. In some sense, an exclusion is absolute. There's no need to talk about what the chance that some other person might be excluded. He's just excluded, and we can leave it at that and move on. So when we move on, what do we need to do? Well, once again, we're going to need to do some comparisons between the evidence sample and another suspect's sample. And so the evidence sample here hasn't changed at all, but now we're going to consider a different suspect. And what do you see when you start doing that same locus by locus comparison we did for suspect number 1? Well, remember at D8, the evidence sample had a 12 and a 15 as part of the DNA profile. And oh, sure enough, at the D8 locus, we see that 12 and 15 showing up in the reference sample from this particular suspect. And at locus after locus that we look at, we're going to see again and again that suspect 2 could well be the source of that DNA. The next locus over, a 28-29, sure enough, a 28-29 in the reference sample. Over here, an 8 and a 12, here an 8 and a 12 as well. And I'll leave it to you to look through the rest of these loci and confirm for your own satisfaction that suspect 1 can't be excluded, but I've already given you the answer. The question is, what can we conclude? I think it's pretty straightforward, and again, there's nothing really deep thought that's going into this. Suspect number 2 cannot be excluded as a possible source of this evidence sample. All right, so there's an interesting philosophical aspect to what's just transpired here. Notice for both of those comparisons, the first thing that we were trying to do is see if the suspect, the individual that we were comparing to an evidence sample, could be excluded. That's the first objective. It's when we fail to exclude an individual that then we have to start talking about how impressed we should be by that failure to exclude. In some sense, how many other people might we have to look at before we would similarly not be able to exclude them? In other words, find somebody else who might also be a possible contributor to that evidence sample. And so that is really the question at hand for this particular video. What weight do we want to attach to DNA profile matches? When we find that an evidence sample and a reference sample have in fact the same profiles, that the source of the reference sample can't be excluded as the source of the evidence sample, we need to be able to attach a statistical weight. And let me say a few words just briefly about statistics in general. There's this popular expression that's there's lies, damn lies in statistics, right? Well, I want to make for the record here the fact that statistics themselves aren't actually lying. But the thing is we have to be really careful, and I think I can't emphasize that enough. We have to be extremely careful that we're paying close attention to the question that a statistic is attempting to provide an answer to. So what is the question that we're worried about here in this context for DNA profiling? Well, right now we're talking about what's the chance that a randomly chosen, unrelated person might happen to have the same DNA profile that we find associated with an evidence sample, all right? In a nutshell, that particular question is known in forensic DNA profiling circles as the random match probability, or often abbreviated just RMP. And again, I have on that slide the specific language associated with the random match probability. It's the chance that a randomly chosen, unrelated person might have, from a given population, might have the same DNA profile we see in an evidence sample. any word in there that gets changed is going to have an important implication to the overall statistic that we calculate. So bear that in mind. But for now, we're going to talk about the simplest case scenario where those words are precisely the words that we have in the question that we're addressing. So how does that get done? Well, in some sense, it's accomplished by an application of a statistical principle known as the product rule. And what I'd like to do now is just talk with you about how it is that the math that underlies the product rule comes into play as we're generating these statistical weights. So again, this is for a single source sample, no mixtures, all the information that's been contributed by the source of the DNA has been assessed and we have it in hand, we know what's happening throughout the sample. The formula that's involved for calculating the statistic, this random match probability, is pretty straightforward. And it needs to be broken down into two different components. There's some loci where we're going to see two peaks in the evidence sample and the reference sample, these matching samples. Those loci where we see two peaks, we say that at that locus, the individual is a heterozygote, right? And quite simply, that means that we've observed two peaks there. There are other loci that we'll see as we're looking at an electropharogram where we might find just one peak. In that case, what's happened is we call this type of individual a homozygote at that particular locus. And what it means, as I've alluded to already, is that individual actually has two copies of that particular version of that locus, two copies of that allele. Here, the individual has two copies of the 9.3. If you're curious, what that means is that the individual's mother and father both contributed a 9.3 to him as he was developing as an embryo. But at some other locus, like the D8 locus, his mother may have given the 12 and his father may have given the 15, or vice versa. All right, so that's the essence of what's going on here. There's two kinds of loci. Loci where the individual is a heterozygote because they have two peaks, and loci where the individual is a homozygote because they have one peak. We're going to use two different math formulas for the heterozygous loci and the homozygous loci. Let's talk about heterozygous loci first. At those loci, the formula that's going to be used is simply 2 times P times Q. Okay? So at any locus where the individual is a heterozygote, we're going to invoke 2 times P times Q. You're probably wondering, what are P, what are Q? There's nothing really magical or mystical about them. P is the frequency of one of the alleles that we find in that particular profile. Here, for instance, we'd wonder what's the frequency within some population of the 12 allele at the D8 locus. We can plug that frequency in place of P in this formula. Similarly, Q, you probably don't even need me to tell you this, right? Q is the frequency of the 15 allele at the D8 locus. And so if we want to know how common or rare this particular combination of alleles are, all that we need to do is determine the frequency of the 12 allele and the 15 allele, multiply the two of them together, and multiply that value by 2. Okay? And we'll do that for all of the heterozygous loci. Any locus where there were two peaks, that same formula is going to apply. And that again is in contrast to the 3 loci that you'll see here, where the suspect and the evidence sample are homozygous. And again, there we're going to use a different formula. There it's simply P squared. There's nothing surprising about that. The formula pretty much says it all. All that we're going to do here is multiply the frequency of the 9.3 allele times the frequency of the 9.3 allele. And we'll do the same thing for all of the other homozygous loci. And when all is said and done, what's going to happen is when we want to talk about how common or rare a combination of alleles at a locus is, that's the formula that we would use at each individual locus. But at the end, we're going to want to know how common or rare the entire combination of alleles are. And to do that, all that we'll need to do is multiply the answer that we got for each individual locus times the value we got for all of the other loci for which we have information. So we're going to multiply them all together, and that is going to give us the random match probability. Okay? Remember, the random match probability is telling us what's the chance that a randomly chosen individual from a given reference population would have the same combination of alleles that we see in this suspect and in this evidence sample across all of the loci for which we have information. All right? And the way we're going to get there is simply by multiplying 2 times p times q for heterozygous loci and p squared for homozygous loci. So let's put this abstract P and Q business to the test and see what we do in a real world scenario, right? What's the chance that a randomly chosen person would have the same DNA profile that we've been talking about for the last few minutes? Well, how are we going to assess that? First, we need to figure out frequencies for the 12 allele and the 13 allele at the D8 locus. Back in the late 1980s, the FBI, the Federal Bureau of Investigation, anticipated that there would be a need to do these types of calculations. And what they did is they took about in the ballpark of 200 FBI recruits coming to the FBI Special Agent Academy in Quantico, Virginia, at the end of the 1980s. They took 200 of those recruits and they asked them for some blood samples and generated DNA profiles from them. And those DNA profiles have in turn been used to tell us how common and rare many of these alleles are that we'll be talking about today and in subsequent videos. And so, what did they find? Well, when they looked at their 200 or so FBI recruits, here they had 196 for which they were able to get information at the D9 locus. What they found is that 14.5% of those 196 individuals had a 12 allele. So what's the frequency of the 12 allele? Within the Caucasian population, again, these were 196 Caucasians here, within that Caucasian population, 14.5% had the 12 allele. That is the frequency of the 12 allele. Now similarly, we're going to need to figure out the frequency of the 15 allele. And we can look back to that same population and see what frequency the 15 allele occurred in within Caucasian individuals, those 196 individuals. And what you'll see is that just a little bit less than 11% of those 196 individuals had a 15 allele. So, I feel almost like this is a pop quiz. What's the chance that you might find in a Caucasian population an individual who at the D8 locus has both a 12 and a 15 allele? You're going to need to invoke that 2pq formula. You've got p here, you've got q there. You need to multiply by 2. Strictly speaking, technically it's because the 12 could have come from the mother or the father, and the 15 could have come from the mother or the father. There are 2 ways you can become a 12-15. But when you do that math, and calculators can do that easily, when you do that math, you get a value of 3.2%. So remember, my question was, what's the chance that a randomly chosen, unrelated Caucasian would have a 12 and a 15 at the D8 locus? The answer to that question, 3.2% chance. That means in some sense that you'd have, if you looked at 100 Caucasians, you might expect to find 3 of them with a 12-15 combination of alleles at the D8 locus, just like what we see in this evidence and this reference sample. Okay? Now, of course, we're not going to stop at just the D8 locus. There are many other loci that can be looked at here, and we'll do the same exercise for all of them, but I'll leave it to you to do the math if you care to. And once we have all these values, remember the last step in this product rule business is we're going to multiply all of those frequencies together and look at the number that we get, right? Impressed, I hope, right? That is a big number. In fact, that's such a large number that I think it's better than talking about all the zeros, counting the zeros and so forth. Let's just put a word on it. When mathematicians talk about a number with that many zeros, they're talking about quintillions. And more specifically, that particular random match probability for this DNA profile is 1 in 609 quintillion. That's billions of billions, right? And when I said at the beginning of this video that 13 or here a 15 locus DNA profile match is exceedingly rare, that is what I meant by exceedingly rare. 1 in 609 quintillion kind of rare. Think again about what that statistic means. That statistic means that we'd probably have to look in the ballpark of 609 quintillion individuals before we found another one that we expected to have a similar DNA profile, the same DNA profile that we found between that evidence sample and that suspect. Exceedingly unlikely to happen by chance. Now, let's get with that under our belt, right? I mean, that's an important lesson, right? There's something important to be said there when we're talking about this rare of a match. Let's talk about what's under the hood a little bit more about the product rule, because there are actually two assumptions that underlie what it is that we've just done by applying the product rule in this way. Okay? The first of those assumptions is this one that needs a little bit of explaining, right? The first assumption is that the events that we're talking about here, the presence of a 12 allele and a 15 allele, for instance, at the D8 locus, that those events are independent of each other. Now let me put this to you plainly. What that means is that because we see the 12 allele at the D8 locus, we're not any more or less likely to see some other allele at that locus. In other words, just because you have a 12 doesn't mean you can't have a 14. Or just because you have a 12 doesn't mean you have to have a 15. They need to be independent. And in addition to that, not only do these alleles need to be independent within a locus, there's a need for independence across loci as well. If that assumption isn't true, that there's this independence of the presence or absence of alleles within and between loci, then we don't have any business multiplying those allele frequencies together. Well, we're starting to stray into a realm here of a discipline known as population genetics. It's population genetics that we can turn to help us answer that question: are these alleles independent of each other or are they not? And within population genetics, there are actually two specific tests that can be applied to answer those questions. To answer the question of independence within loci, population geneticists will turn to what's known as Hardy-Weinberg equilibrium. And Hardy-Weinberg equilibrium is actually the cornerstone of all of population genetics. There's some fairly elegant but also straightforward math associated with the Hardy-Weinberg equilibrium, but we don't need to get into that for the purposes of this video and what we're trying to do with these DNA profiles. All that we really need to establish is that there is a test that we can use to see if that first assumption is true, that alleles are independent of each other within a locus. And for that, again, we can perform the Hardy-Weinberg equilibrium test, and if we get a positive answer, we can come away with the understanding that it might not be unreasonable to multiply together those allele frequencies as we've been doing for the past few minutes. And just as Hardy-Weinberg equilibrium is something we look for independence within a locus, we can look to a separate test called a linkage equilibrium test to see if there's independence across loci. All right? If both of these tests come back and tell us that the alleles that we're talking about, the populations of individuals we're talking about, have no associations within loci or between loci, we can use the product rule, that multiplication, without any concerns. If, however, these two tests or one of these two tests tells us that there are some dependencies, right? The fact that one allele shows up in conjunction with another allele more common than we might have expected by chance. If there are those dependencies, the good news is that population genetics also gives us some tools to compensate for those dependencies. In short, I can talk with you in some depth if we want to get into those details in some subsequent video about a theta correction that can take into account the fact that there are some dependencies. But for all intents and purposes, those theta corrections and the corrections that are used to account for population substructure, if and when they're found, tend to have relatively little effect on the overall bottom line number. Practically speaking, that 1 in 609 quintillion number that came up a couple slides ago, when you factor in population, genetics, substructure considerations, and you use a theta correction, that 1 in 609 quintillion number might be downgraded to something like 1 in 509 quintillion. The bottom line answer is still pretty much the same. It's an exceedingly unlikely thing to find two individuals with perfectly matching 13, 14, 15, or 16 locus DNA profiles. Those sorts of things are just very unlikely to occur by chance. So, there were two assumptions that underlie the use of the product rule. The first one that I've just talked about is this idea of independence within and between the alleles that we've detected in those electropherograms. The second one is one that's almost embarrassing to mention. I think to some extent it goes without saying, but if it doesn't get said, people seem to have a tendency to forget it, and that is that we do need to know what the frequencies of those alleles are in the first place, right? So we really need to know how common or rare a 12 allele is at the D8 locus within the population that we're considering. And so we've already talked about how it is that the FBI got an answer to that question in the context of Caucasians, but let's take a careful look at that, because during the course of this video, as you've been watching it, some of you may have already had some questions about, gee, is 200 individuals enough to get that sort of estimate? And are 200 Caucasians enough to get that sort of estimate? So let's look back then at that random match probability definition. Remember, that's the key here. We got to keep our eye on the question that this statistic is intended to address. The question that we're trying to address with a random match probability is this: what's the chance that a randomly chosen, unrelated individual from a given population would have the same DNA profile that we've observed? And let's focus our attention right now on that one little clause in there about from a given population. What population are we talking about? Well, for the previous couple of slides, we were talking about Caucasians. And why might we consider Caucasians? Well, I'll tell you why we might consider Caucasians. The key here is that in many circumstances, for crimes that are committed within the United States, Caucasians are a fairly good way to describe a pool of alternative suspects to the individual that we've been investigating, to the individual whose DNA profile we have found matches that in an evidence sample. And I think that is a key here. They are a pool of alternative suspects. And what we need to do with the random match probability and the calculation we've just performed is essentially rule out of consideration this wide swath of the population in the United States. We're saying we've got an individual whose DNA profile matches the evidence. What's the chance that some other person might match? Well, what other people might we consider? A big pool of people in the U.S. that we might consider would be Caucasians. When we say the chance that a randomly chosen unrelated Caucasian would have a 1 in 609 quintillion chance of matching the evidence sample the way that the suspect does, we can pretty effectively eliminate from consideration the possibility that some randomly chosen Caucasian might be the actual perpetrator of the crime, and that we've found this suspect by an unfortunate coincidence. But can we stop there is an important question. Now that we've eliminated from consideration the possibility that Caucasians are the source, with the possible exception of the one that we have in hand, if the suspect is a Caucasian, we also need to start thinking about, are there other pools of suspects that we might want, of alternative suspects that we might want to rule out of consideration? And so for the purposes of DNA profiling statistics, it's very common for a testing laboratory to report at least 3 different statistics. They'll eliminate from consideration the pool of alternative suspects that we call Caucasians. They'll also eliminate from consideration, by use of the random match probability, the pool of alternative suspects that we call African Americans. And they'll do a third calculation where they rule out of consideration the possibility, how likely it is that an alternative suspect might have arisen from a pool of alternative suspects that we'll refer to as Hispanics. So those three large racial groups that constitute the U.S. population are effectively addressed one by one, and again, large swaths of the population simply get ruled out of consideration each time we apply that random match probability statistic. Notice, it's not important for us to know what the ethnic background or the racial background of the suspect is. That doesn't factor into the consideration at all. I don't care if the suspect is African American, Caucasian, or Hispanic. I simply know that he matches, and then now I want to know what's the chance that some other person walking down the streets might similarly match. And to do that, I focus on those three racial groups and get there. And again, never do I have any concerns about what the ethnic group or the racial group of the suspect is. The only time that we might want to consider some other population beyond African Americans, Caucasians, and Hispanics is if we can reasonably believe that the true perpetrator of the crime is not reflected or isn't captured, is not a member of one of those three large racial groups. Asiatic comes to mind, Pacific Islander comes to mind, Eskimo comes to mind, some other Native American population. If we think that there's a reasonable chance that the perpetrator of a particular crime falls into one of those alternative racial or ethnic populations, then what do we need to do? Very simply, we need to figure out the frequency of the alleles in those alternative populations and see if we can't eliminate from consideration those alternative suspect pools as well. So, there's this second assumption. We need to know the frequencies of the alleles that are important to our question, and we need to focus on the population for just a little while longer. In the context of a DNA profile, the populations that frequently get talked about are Caucasians, African Americans, and Hispanics, right? But what I'd like to do is move things out of the realm of DNA profiling because, quite simply, that's something that people feel there's a charge in the air when you're talking about random matches and DNA profiles. Let's make this a little bit more abstract, but in a way that I think you'll see translates very directly to what we're talking about here for DNA profiling. And so for a little while now, instead of talking about DNA profiles and allele frequencies, let's talk about something else that doesn't necessarily give rise to these staggering numbers, but something else that people may well find interesting and maybe help illustrate some other issues and concerns about this matter of what populations we need to pay attention to. And so from this slide, you can tell that I want to change our attention here to talking about presidential elections, right? Bear with me, right? Let's say that for some purpose, and we can come up with a wide variety of reasons why we might care to know the answer to this question, but that for some purpose, it's important for us to know what fraction of individuals who voted in the 2008 presidential election, what fraction of those individuals cast their vote for Barack Obama, right? Again, for whatever reason, let's say that that's an important matter for us to consider. Well, what would be the best way for us to get an answer to that particular question? It turns out it's fairly easy to get a very precise answer to that particular question. North Carolina, like every other state, maintains a record of the answer to that precise question. And across the United States, again, every state has done this due diligence of maintaining those records, and we can go to North Carolina's records and find out that in the 2008 presidential election, just over half of the voters in North Carolina cast their vote for Barack Obama. Let me just say this. That is the right answer to our question, right? If we want to know the frequency, notice how I slipped that word in here. If we want to know the frequency of individuals who cast votes in North Carolina for Barack Obama in the 2008 presidential election, the correct, precise answer is 50.2%. But, let's say that for whatever reason, it turns out that we don't have access to North Carolina's data, right? Our web search didn't yield that specific number. North Carolina's records were compromised in some way. Again, imagine whatever reason you like. Let's say we can't get the ground truth, absolutely correct answer of 50.2%. What might we use as a surrogate? How else might we go about finding that particular frequency? Well, one alternative might be just to ask what fraction of people who cast votes on the eastern seaboard, right, that eastern half of the United States, that region, what fraction of those individuals cast their vote for Barack Obama as opposed to John McCain? Well, I can tell you from looking at election results that for that 2008 election, just under 60% of those who cast votes on the East Coast cast their vote for Barack Obama. Now remember, I'm trying to set this up here so that you think that we want to find the answer to the question, what's the fraction of individuals in North Carolina that voted for Barack Obama? That's the question we want to have answered. We can't get the answer quite the way that we want. I'm tying our hands behind our back in some sense here, but we can get a surrogate answer, and you'll see that it's not that far off. It's not the same, right? Instead of 50.2%, we're seeing the number here is a little bit off, but the general answer is still in the ballpark. We see that more people supported Barack Obama than supported John McCain in casting their votes, and again, maybe that answer will suffice. It's not exactly the same as the right answer, but maybe it's close enough. But let's tie our hands even a little bit tighter behind our backs and say, well, I want to know the fraction of individuals who voted for Barack Obama in North Carolina, but I don't have data from North Carolina, and I can't even break down the data from the perspective of the East Coast. All I can do is look at the overall U.S. population, right? What does that do to our estimates of these frequencies? Well, if you get that particular bit of data, you would find that just under 53% of those who cast votes in the 2008 election voted for Barack Obama. And hey, that's not so bad from the perspective of we want to get the right answer. The right answer, again, is 50.2%. We know that because of the way this question has been set up. And so when you look at a broader population, we're actually getting a number that seems a little bit closer to the right answer. Now, of course, I'm sure you've appreciated that sometimes when you look at a broader region, you'll get a less correct answer. But in this circumstance, maybe using the overall U.S. vote percentages gets us pretty close to where we want to be, knowing what it is that, what fraction of the population voted for Barack Obama. But let me ask you this. So, the best way to get the answers, clearly, just go right to North Carolina. Maybe the next best way is to go to a region, right? The whole East Coast. If you don't have access to that, there may not be too many problems with going to the whole country, right? To a much broader population, right? But let me ask you this. Would you seriously consider getting the answer to that question by not going to North Carolina, not going to the East Coast, not going to the entire U.S., but rather to a state like Utah? Seriously, would that make any sense at all? You want to know the answer to this question, what fraction of individuals in North Carolina voted for Barack Obama? Do you think you're going to get a good answer by not looking at North Carolina, but rather by looking at Utah? I say that somewhat facetiously, but because I think it's pretty obvious that there's a good chance that now we're pretty far afield and we may not get a very good answer. And it happens that when you look at the voting results for the 2008 election, John McCain apparently had quite a bit of support in the state of Utah because only 35.5% of the people who voted there cast their vote for Barack Obama. So do you see where we're going with this? Right? What we're talking about here is there's a frequency that we think is important, right? Let's say the population we're really interested in is North Carolina. The right answer might be something we could best obtain just by going to North Carolina. But if you can't go to North Carolina, what's the next best thing to do? Maybe go to the region that contains North Carolina, maybe broaden the region in one that still encompasses North Carolina. But one of the worst things that you might do is go to a completely different population and ask, what's the frequency, and try and superimpose that on the region or the state that you're interested in. So let me tell you something. What's all this have to do with DNA profiling? Well, again, I think you've probably connected the dots here already, but when we're doing random match probability statistics, one of the things we really need to know is the frequency of the alleles that we're multiplying together. If you want to know the frequency of alleles within a Caucasian population across the entire United States, what's the best way to get that answer? Probably by getting a random sample of all of the Caucasians across the entire United States. You may not have to get every last Caucasian entered into your allele frequency database, but you want to get a random sample distributed pretty much from all over. You know what's happened, though, for the allele frequencies that we work with routinely across the country when we're multiplying numbers together for random match probabilities? More often than not, crime laboratories use an allele frequency database that's derived from the FBI's database, and those 200 or so FBI recruits that came to the FBI Academy back in the late 1980s, upon which those allele frequencies are based, guess what? Studies have shown that most of them were Mormons. And not only were most of them Mormons, they were recruited specifically from Salt Lake City in the state of Utah. Stop and think for a moment about what those implications are. Now, if we want to rule out of consideration the possibility, if our pool of alternative suspects are Mormons from Salt Lake City, Caucasian Mormons more specifically from Salt Lake City, we've got a really good allele frequency database to do that, don't we? Right? That's the one that everybody's been using. But if we're talking about a crime that's occurred in Dayton, Ohio, or a crime that's occurred in New York City, do you think that we really need to worry hard about what's the chance that a stray visiting Caucasian Mormon from Salt Lake has come through and been the perpetrator of the crime? Do we need to go out of our way to eliminate those individuals from the pool of alternative suspects? Perhaps in some crimes that would be an important consideration, but I think it's pretty obvious that for most crimes that's not something that we're going to be particularly worried about. So where are we? Well, where we're at is we have allele frequency databases for Caucasians and for African Americans and for Hispanics that may or may not reflect the population of alternative suspects for where a particular crime has occurred. You can see for the numbers that we had here for the election results back in 2008, for that fairly straightforward question, we can be off by quite a large margin, 15% or so off between what the correct answer was and what the answer that we may have used is. And while 15% may not sound like that much, bear in mind that those types of 15% differences are applying to very large numbers. 15% off of 609 quintillion means that you're in the ballpark of what? 40 or 50 quintillion off. Is that an acceptable margin of error? Maybe, maybe not. Bear in mind this as well. Those errors are multiplicative. What that means is that if you're 10% off with one of those allele frequencies, and you're 10% off with another allele frequency, let's say in both instances overestimating the rarity of those alleles by 10%, when we look at both of them together, we're not off by just 10% or 10 plus 10%, 20%. We're going to be off by 10 times 10%, or a much larger fraction. Right? So we can have some pretty wide variance here in terms of what the right random match probability is relative to the random match probability that's actually being reported. But let's zoom back out here a little bit and look at the big picture. Again, a 13 or a 14 or a 15 or a 16 locus DNA profile match is exceedingly rare. All right? And the things that I've talked with you about now for the past 10 or 15, maybe 20 minutes, might make you think that, gee, the random match probability has some serious problems associated with it, right? In some sense it does, but it doesn't change that first bullet on this slide, and that is that 13 and more locus DNA profiles, matching DNA profiles, are extremely rare. How rare might be something that we're going to be debating, right? We can quibble over, is the number 609 quintillion or is it 509 quintillion? Maybe if we factor into account all of these things, the population matters, the correlations between and within loci, maybe the numbers actually is as unimpressive as 1 in 100 billion, right? But think, seriously, 1 in 100 billion, that's telling me pretty much the same answer that 1 in 609 quintillion was telling me. That's telling me that the individual that we've found that matches the evidence sample's DNA profile is virtually certain, to the exclusion of all others, the source of that DNA profile, right? The numbers may be off a little bit, but the bottom line stays the same. That's what we do when we have a single source DNA profile where we have complete information and we're confident that we've gotten true, reliable, accurate results, right? The statistics can be staggeringly impressive. So staggeringly impressive that few defense attorneys these days bother to go through the quibbling about the relevant population or to go about the quibbling about the population substructure that we might find with Hardy-Weinberger linkage equilibrium. But the story doesn't necessarily end there, right? That is the best case scenario. And I'd like at this time to encourage you to watch another video in this series where we talk about how things might get a little bit more complicated, because many cases do involve single source samples that look as if they've come from a reference. But many evidence samples have problems associated with them. Things such as the fact that they constitute a mixture, or we don't have the complete story, or close relatives might have been contributors and we need to rule out the possibility that a sibling, for instance, was the source of the DNA that we're seeing. Those sorts of things invariably work against making those statistics impressive. They invariably make the statistical weight that gets attached to a DNA profile match less than it would have been if they hadn't been in play. When they're not in play, random match probability statistics in the quintillions are very common. When they are in play, then we can start talking about, in some instances, random match probabilities or some other sort of weight that might be described in 1 in just a few hundred or 1 in just a few thousand. In those circumstances, the problems associated with the allele frequency databases and dependencies, then they may actually start to take on a real significance, and there's something that we might want to take more careful consideration of. But for a straightforward case where there's nothing but a DNA profile that looks like an evidence, like a reference sample, where the evidence sample looks like a reference sample, and where we're only worried about randomly chosen, unrelated individuals being an alternative source of the DNA, the random match probability is the way to go, and we probably don't need to worry too much about those concerns associated with Hardy-Weinberg and linkage equilibrium and concerns associated with the samplings of the database. So that's what I wanted to talk with you about in this video, about how it is that we calculate statistical weights for single source DNA profile matches. Again, what you'll find in a subsequent video is we'll be talking about some of the things that make this a little bit more complicated. That's the simple case, best case scenario for calculating a statistic. Other videos will talk with you about how it is we deal with the problems that get raised by mixtures and other problems such as degradation and inhibition and distinguishing between signal and noise and technical artifacts and things of that nature. So I'd encourage you to come back and take a look at those videos, but for now let's leave it at that, and that's how it is we calculate statistical weights for single source DNA profile matches.